Hello students, I am Dr. Salahuddin from Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Today I will discuss about the various approaches in the drug design. So, first of all I will tell you about the drug and what is the drug design. So, the drug is most commonly organic molecule that activates inhibit the function of a biomolecule such as protein, which in turn result in therapeutic benefits to the patient. Now, our aim in the drug design is to produce a newer molecule, and in the drug design it may be defined as the effort to develop a new molecule. Why we are developing a new molecule? Because in today's scenario there is a need to develop a new molecule for the various types of diseases, and as a medicinal chemist our role is to develop a new molecule with the help of the modification of the lead compound. So, here there is a one term, that is the lead compound. First of all, what is the lead compound? So, the lead compound here, lead compound means the compound, the first compound which shows the biological activity towards the receptor, and after the modification of this lead compound there is some properties that we have to discuss in the drug design part. Now, here this is the complete drug discovery cycle, and here the first thing is the compound collection. So, our first aim is to collection of the compound, and here the collection of the compounds from the different types of libraries we can collect, and the different chemical synthesis that we can involve in this to produce the compound collection. Now, after the compound collection there is a various types of the compound collection. So, that there is a it produces the library, and so the screening is required at this step. So, the primary assay with the help of high throughput screening, and it is a in vitro screening. With the help of in vitro screening of the compounds, and it is a we can screen a number of the compounds with the help of high throughput screening, so that the number of compound will be reduced. And now come to this secondary assay. With the help of this secondary assay we can observe the efficacy of the compound, we can observe the bioavailability of the compound, we can observe the toxicity of the compound, and in some cases we require the in vivo screening. So, that after the primary assay and the secondary assay we will get the candidate drug, or sometimes this one is lead compound. So, this lead compound, after the finding of this lead compound we have to develop the structure activity relationship between the compounds. So, so here for the structure activity relationship of the lead compound we have to modify the compound. After the modification of the compound, and then observe the activity of the compound and develop a correlation between the biological activity and the structure. So, that the structure activity relationship will establish, and after that structural characterization of the protein ligand complex. So, in the drug discovery cycle and in the protein drug designing we can observe the complex between the receptor and the ligand, so that there is a complex will obtain, and after the formation of the complex, we have to characterize is which type of the protein ligand complex formed and what is the characteristics of this protein ligand complex and which type of bond formation occurs in the protein ligand complex, and this will lead to a design of a molecule. Design means our requirement. On the basis of our requirement, we have to design our molecule. Without doing all these phenomena, we cannot design a new molecule, and after that we have to synthesize the compound. This is very important step, the synthesis of the compounds. Now this computer aided drug design is used for rapid assessment of the chemical libraries in order to guide the speed of the early stage development. The early stage development means what we are doing after the identification of the disease. If the disease is new, that we have to develop a molecule against the particular disease. So, it is a early stage development. Now, the drug design helps the scientist in the minimizing the synthetic and biological testing effort. So, what is this minimizing the biological testing effort? So, in the conventional drug design, that we are synthesizing a number of molecule. So, after the synthesis, we are doing the testing of all the compounds. Now, here with the help of drug design, the number of compounds will be less. So, and we have to focus only the most promising compound. Most promising compound means the compound which shows the very good biological activity. And besides explaining the molecular basis of the therapeutic activity, it also predicts the possible derivatives that would improve the activity, that is called the structure activity relationship, or we can say that is the optimization of the molecule. Now, in this computer aided drug design, there is a four important steps required. That is, first step is ligand, or we can say the drug selection or designing. What we can do in this, we have to do the database mining. Database mining is what? Whatever I am going to perform any type of study, first of all we have to thorough study about this. The database mining, we have to observe the number of compounds already developed, number of receptors already developed here, so that it is a database mining is very much required for the designing of any type of molecule. Now, next thing is structure similarity. What type of structural similarity of the compounds that is previously described molecule. After the identification of the ligand and number of ligands, we have to predict the target. Now, here there is one problem arises where we can predict the target and how we can predict the target molecule. So, nowadays it is very simple that we can predict the target structure with the help of in silico tools. In silico tools means the various tools are nowadays is available where we can predict the structure of target. Now, various types of pathway involved in that particular target. Now, what is this? That this protein that we have already choose, what are the various pathway involved? What the biochemical phenomenon occurs in this? So, after the pathway analysis and we have to structure modeling of the target. Third thing is ligand target interaction. Third and very important step here, ligand target interaction, and with in this ligand target interaction of the study of the ligand with the target, we can observe with the help of docking. So, docking is what? Docking is the interaction of the ligand ligand with the receptor, and it will forms a complex. With the help of docking study, we can observe which type of the interactions occurs, which type of bond formation occurs in between the ligand and the target. Okay. And next thing is virtual screening. If we have a large number of compounds, so that we cannot do all the interactions with all the molecules with the target, we have to screen on the basis of our requirement. First, we have to choose the pre-requirement of our study, so that we can observe the interactions of the compounds, and we can observe, or we can filter the compounds with the help of this virtual screening. Next an important thing is Qacr analysis. Qacr, quantitative structure activity relationship. We have to establish the quantitative structure activity relationship. So, the definition says of the Qacr is what? It is a mathematical relationship between the biological activity and the compound. So, that we have to establish the relationship between the biological activity and the compound which we have already selected for our study. So, and the next thing is preclinical testing. In the preclinical testing, we have to do ADME analysis, absorption, distribution, metabolism, execution, and the toxicity study. So, nowadays various types of in silico tools are available that we can observe the ADME analysis. Next thing is Lipinski rule, and in this Lipinski rule we have to observe the molecular weight of the compound, we have to observe the hydrogen bonding acceptor of the molecule and the hydrogen bonding donor capacity of the molecule, and the last one is the log p value of the structure. So, that there is some limitation in this Lipinski rule. So, with the help of this Lipinski rule, we can observe the molecule, either this molecule may act as a drug molecule or not. Now, next thing is drug likeness. In the drug likeness, on the basis of the analysis, that is ADME analysis, Lipinski rule and the toxicity analysis, then we can say that it is a drug like structure in the drug design. Now, in the drug design and discovery, we have ligand based approach and the target based approach. So, here the drug design we can do with the help of the ligand based and the target based. So, what is the basic difference between the ligand based approach and the target based approach? So, in the ligand based approach and the target based approach, in the ligand based approach our ligand is known, and the target based approach in the target based our receptor is known, our proteinaceous protein is known. So, when the protein is known, if we are doing on the basis of the known target, that is called target based approach, and it is more rationalized towards the drug design. And first I will discuss about this ligand based approach. In the ligand based approach, first thing is, first we have to identify the pharmacophore. Now, here what is the pharmacophore? the basis of identifying of this pharmacophore is the basic moiety which is responsible for the biological activity. And in this pharmacophore, first we have to identify the basic structure present in the series of the compound, and we on the basis of this that we can observe the pharmacophoric structure of the compound. So, in this pharmacophore identification means we have to identify the basic characteristics of the moiety. So, after the identification of the basic characteristics, we can identify this pharmacophore, and if there is any lacking in this pharmacophore, then we have to develop the modification. We have to do the modification in this particular structure, the basic structure we can do. So, our aim is to identify the pharmacophore and then modify the pharmacophore on the basis of our requirement. Okay? And after the identification and the modification of the pharmacophore, we have to do the docking with the receptor and what dock to the receptor. So, our ligand is known, but the target is unknown here, because I am doing this ligand based approach. So, we have to choose the target for this particular ligand of our identified pharmacophore, and then we have to identify the potential drug or the potential lead compound in this ligand based drug design or the ligand based approaches of the drug design. Next, now come to the target based approach of the rational drug design here. So, here we can do the docking approach. In the docking, we know the receptor, and in the receptor we have to identify the ligands against the known receptor. If our receptor have hydrogen bond like hydrogen bonding acceptor, so in our ligand it must have the hydrogen bonding donor. Okay? So, there is a correlation between the target and the ligand. Now, next thing is active site binding pocket identification. In this target based, we can identify the active sites present in our protein with the help of softwares, and we can do the 3D QSR, the virtual screening approach. In the target based approach, we have to identify this molecule ligand or the fragment growing. We can this is the another approach of the target based drug design or the structure based drug design. Now, here we can, if it is no, then we can change the fragment. Okay. Now, here this is the simplified diagram of the ligand based drug design and the structure based drug design. So, here in the ligand based drug design, as I have already given the concept of this ligand based drug design, where the ligand is known. So, in the ligand based pharmacophore, we have to identify the ligand, and after the identification of the ligand, we have to identify the pharmacophore, and then we have to do the we can do the 3D QSR, COMFA and COMSIA we can do with the help of 3D COMFA and COMSIA. Now, next thing is 2D similarity based approach, scaffold hoping. This is more important, scaffold hoping, and this ADME prediction in this ligand based pharmacophore design. Next is structure based drug design. In this structure based drug design, we have to do druggability prediction. So, what the drug do against the specified target. So, the target we know about the target in the structure based drug design, and we have to identify the druggability prediction. And this docking based virtual screening, docking based virtual screening means screening of the ligands against the particular target. We have to identify identify the ligands, and with the help of identified target we can do the docking based virtual screening. Next is structure based. In the structure based pharmacophore, next we can do the molecular dynamics also and the de novo drug design. So, in the ligand based drug design, we indirect drug design also we can see the in the ligand based. Structure based drug design, that is direct. Okay. Direct drug design and the indirect drug design. So, we can observe the interaction of the ligands with the target either indirectly or the directly, that is called ligand based and the structure based drug design. And here there relies the knowledge of the other molecule that binds to a particular biological target, is the use of the design of the new molecule. Next is this other molecule may be used to derive the pharmacophore model that define the minimum necessary structural characteristics. We cannot observe all the characteristics of the molecule, we have to minimize the characteristics of the molecule. And here next is ligand based pharmacophore modeling. In the ligand based pharmacophore modeling, we can do the lead optimization and the de novo drug design. De novo means start afresh. We can start a new compound. We do not know about anything. So, that is called the de novo drug design here. Now, in the Qacr, in the ligand based drug design also it includes the Qacr. So, Qacr quantitative structure activity relationship, that this is the method of the ligand based drug design, the important for the prediction of the biological activity against biological activity or of the chemical compounds based on mathematical and the statistical relations. So, we have to establish the correlation between the biological activity and between the biological activity with the compound structures. So, we have to establish on the basis of the mathematical manner or and the statistical correlation between these two, and this Qacr approach determine the physical properties based on molecular features of the various compounds. So, here we can do the 2D similarity based, we can observe the structural representation of the molecule in the ligand based, quantitative measurement of the similarity between the structure representation, and it is based on the two molecule often share with physical properties. Similar principle widely utilized for the development of the new molecule. And next is scaffold hoping. In the scaffold hoping as an approach to generate a new chemistry, start from any lead structure. So, in the scaffold hoping we require at least one lead molecule, and on the basis of this we can apply the method of the drug design by altering the replacement of the central core of the known bioactive molecule. It may be similar or it may be dissimilar, and in the scaffold hoping we have the new lead compound, and altering in the lead compound we have to develop a new ligands and develop the structure activity relationship. In the ligand based drug design, we can do the Admet prediction, and for this Admet prediction it is used at many stages of the drug design, and it is a very good tool to identify the absorption, distribution, metabolism, excretion and the toxicity of the compounds in our study, and we can develop a library with the help of these prediction. Okay. And we can use the tools, that is Swiss ADME, pre-ADME, mole expansion and the ADME modeler. And now come to the structure based drug design. It is a direct drug design. As I have already told you, with the help of this we have to find out the receptor, and after the finding of the receptor we can do the formation of the complex with the help of binding. Now, here the docking is the very best example of the structure based drug design, where we can observe the interaction of a receptor with this ligand, and it will forms a complex. So that we have to study about this complex with the help of the interaction of the ligands with the receptor, and we can observe the potential drug candidate. So that here our ligands we can obtain with the help of the synthesis, we can observe the virtual screening, and the structure we can observe with the help of homology modeling and the structure biology. So, in the structure based drug design we can do the molecular dynamics in a computer simulated approach for the studying of the atoms and molecule in the physical motion, and the atoms and the molecules are allowed to interact for a certain amount of time, providing a picture of the system's dynamic evolution. So, here this is the references for our study. Thank you so much.